So I'm from Sophion, where I'm working with the systems you've probably all seen here in the hallway, the automated patch clamp systems. And my part as a research scientist is to develop more advanced biology to be run on these platforms. And today I would like to talk about neurons. I'm not on. Sorry. It's on now. So today I would like to talk about neurons and models run on these platforms. And I'll be talking about IPC neurons and on primary neurons. And I added to the title here that we are moving towards more physiological assays. And why is this necessary? We all know that, or at least we believe we know that neuroscience is notoriously difficult to work with. But I wanted to show some numbers on this. And found this Nature review where they looked into therapeutic programs. What are the chances that are producing a candidate that would be approved by the FDA? And across all therapeutic areas, neuroscience has the lowest chance of succeeding and get a FDA approval. And it's believed that it's because we're lacking good models. It's neuroscience is complicated and our models are not good enough. So looking into this from a high throughput patch clamp point of view, we have been working with neurological expression system for the last 2 decades. But I wanted to include more physiological relevant cells like primary neurons and IPC neurons. Primary neurons, they are difficult to get from humans, so they are often derived from animals. But that will help the translation into animal models and to a lesser extent into clinical trials, whereas IPC neurons, they are human-derived, so therefore they can help the translation into humans, thereby bridging the translational gap. So, but how are neurons in the automated patch clamp? Because to run the neurons in the automated patch clamp, we need to have them in suspension. So the neurons are deprived from their dendrites, from their network, and you can say on the good side, it's round, so this is a perfect shape for a Walters clamp. And also you have access to the extracellular control over the extracellular environment, so that you can do fast drug applications. And then when you look at our systems, then on the with this setup on the QPatch compact, then you can run 8 neurons in parallel. On the QPatch 2, you can run 48 neurons in parallel, and then on the Qube, you can run 384 neurons in parallel. So this is a high throughput we're looking for, and then with the relevance of the neurons. But I want to show you what is going on in our lab. I brought 3 short stories, and I hope to underscore why, how we can make these models more physiological relevant. And I will start with a study on IPC-derived spinal muscular atrophy diseased cells. Is a collaboration we have with Brain Cell. So they derive these IPC cells, and they get them both from a healthy individual and also from patients. And in this case, patients that are suffering from spinal muscular atrophy. In short, I don't want to go too much into it, but just to remind you that these polyclonal stem cells are derived from humans. So if they're taken from a patient, then the genotype you'll also find in the IPCs. And also when you differentiate them into motor neurons, then they will share the genotype with the patient, and to an extent also the phenotype of the patient, which in this case is a hyperactivity and a decreased sodium channel activity. And then this can be used for disease modeling and also for screening, and later on also toxicity tests. The disease, in short, most of the patients are having a mutation in the SMN1 gene, and this is leading to the depletion of a survival protein, and this will disturb a lot of intracellular processes. So not an ion channel target, but still there's an electrical phenotype of these cells, before they then will degenerate. There is now a treatment available, and this is this compound SMN-C3. It's also known as Risdiplam, and this compound is supporting alternative splicing of another gene, SMN2, which can then produce a functional SM1 gene. But back to our lab, we wanted to see if we could characterize this model. Sorry, and you see that we got the cells from a brain cell already matured, and then we have them in culture for 1 to 2 weeks, and you see this really extensive network that has been formed. And so in real life, this is actually one of our major issues, is to get these cells into suspension. So lift them off the culture dish and then still keeping them alive. We have been through different harvesting protocols, but we now have a catalog of different methods so that we can get these cells into suspension, and they are also semi-healthy. And then we can run them on QPatch and on the Qube. You see here on the Qube, we can also run several different clones at the same time, and this is something we will apply here. This is a plate view, where you can see this is from one Qube run, and each well is represented as a small square, and all the green one are cells that survived. So here you have 384 experiments in parallel. Each represent a unique IPC neuron. And then we can test the neurons using Waltz clamp. We can characterize their sodium channel currents and potassium currents. And then using current clamp, we can see their action potential. So even though that these cells have been deprived from axons and from dendrites, it looks like they don't care, and they still fire action potentials. But then we want to characterize this disease model, and you see how we test the cells from a control, so a healthy individual, and then also from a patient carrying the SMA mutant. And looking at the sodium current, you can see that here in black is the control individual, and in the SMA patients, you see this increased sodium channel activity leading to the hyperactivity of the cells. Then we also had some cells that have been grown together with the SMN-C3 compound, and this study shows that this compound can restore the function of these neurons, or preserve the function of these neurons. It needs to be there all the time when we're growing them. So again, I think this underscores that is giving more physiological relevance to run these IPC neurons, because this is not an ion channel target. It's the drug is not targeting any ion channels, and still we get translational value. We are continuing this study, and we want to look more into the action potential phenotype. But moving on to another cell type, the primary neurons, we get them from rodents. And there we have the same issue, that we need to get them into suspension. And on top of that, in the brain it's a mixed population, so we also need to isolate the neuronal population. And we have tried 2 different methods, either manually extracting the tissue and dissociating it, and then through culture we can enrich the neuronal population, and then again get them into suspension and run them on the systems. Another method we've been using, we have a collaboration with Miltenyi Biotech, who has an automated tissue dissociator, so we get all the cells into suspension. And thereafter, we can label the cells with the magnetic beads and thereby purify the neuronal population and then bring them onto the system. And using the later method, you can see how we can get we can characterize the potassium currents, and we can characterize the sodium currents. And then again, Also, these neurons, they didn't realize they were taken out of the brain, so they also fire action potential. So we could look into action potential morphology and excitability of these neurons. So, but I want to show you something that is currently happening in our lab, and we are so lucky to collaborate with Konstantina Bampali. She's sitting here in the audience today as well, and she is working at University of Copenhagen, where she's a postdoc, and she's working in the Petrine Wellendorf group. She's also having a poster, so I can only encourage you to go and see her later on and learn more. And she is looking at extrasynaptic GABA receptors. And luckily, Jeremy Lampert gave a fantastic talk yesterday. So we all know that besides this synaptic activity of GABA, then there's also extrasynaptic GABA receptors. And they give rise to this tonic current. So it is the one you see down here, and it's most visible when you then block it with an antagonist, and you can see what is the size of this current. And where Lambert showed yesterday that they could be modulated by neurosteroids, the Wellendorf group have shown that it can also be modulated by kinase activity. So this red trace here represents the one I recorded down here. This is in manual patch. And by lowering the kinase activity, then you can increase this tonic inhibition. And one of the kinases that are involved in this modulation is CAM kinase 2, calmodulin protein kinase 2, this guy down here. And this is also the target of GHB. And GHB is a GABA metabolite. It is it's present in the brain, but you probably know it better under the name liquid ecstasy. So it's a club drug. It's also often used as a date rape drug. Luckily, this is not why Konstantina is interested in this drug. She's more looking into the neuroprotective effect of this drug. So she came to Sophion, and we started looking at HEC cells expressing a extrasynaptic GABA receptor, and to see the effect of GHB. So I marked it here. And as expected, there was little or no effect of GHB in this system, because we simply lack the relevance. We don't have the kinase available in these cells. So we need to go into a more relevant model, and she started bringing primary hippocampal neurons. I explained the method earlier, but using this method, we could get cells into suspension, and these are really recent data, so please bear over with the quality. But again, we could characterize the different ion channels in the cells. We could measure action potential, and what made Consti happy is that we can also now look at GABA-mediated current in these cells. So now we can start to evaluate the kinase activity and how we can modulate that. But again, please go and see Consti at the poster session, so if you want to know more about this. I would also like to jump to another study that is currently happening in our lab. We have a visitor. It's a Tabea Klasvausev is visiting us all the way from Australia, where she's working in Irene Vedder's group. And she brought with her nettle toxins that she wants to investigate. And you probably all tried to be stung by a stinging nettle when you're walking through the woods with bare legs in the summer, and know this itchy and painful sensation. But in Australia, everything has to be more toxic, and for some reason. So you can see how the stinging nettles have grown into trees called gimpy trees, and there are warnings against them because it is super painful. You can see here is pictures of a person stung by a stinging nettle. and it can last for hours, days. For some people, this pain lasts for weeks. And the group showed in animal models that the toxin, so they isolated the toxin, is called exaltatoxin, and this toxin, when injected into mice, the pain can be relieved by TTX. So this point is in the direction of sodium channels, and indeed, when looking at primary DRG neurons, so this is manual patch clamp, you can see the sodium current under controlled conditions, and then when exposed to the exaltatoxin, this sodium current inactivation is disappearing, and there's a persistent current causing the pain. The same is also seen in IPC-derived sensory neurons. There's also a loss of inactivation and a persistent current. And the same is also true in TE cells, which are neuronal-derived cells. They then want to see which ion channel subtype or which sodium channel subtype is being targeted by this toxin, because it must be one of the sodium channels. So they expressed the sodium channels in HEC cells, and they saw nothing. And even some other neuronal-derived cell lines, they saw nothing. So this shows that this model simply lacks relevance, so there's something missing in this study. This for the group was lucky because it led to the discovery of a novel protein interacting with the sodium channel, TMEM233. And this is also why Tabea is visiting us at the moment, so that she can use our Qube to evaluate this protein and the exaltatoxin. But Tabea is also sitting here today, and she's bringing a poster, so please pass by her and hear more about the exaltatoxin. So to sum up, I've showed our work on primary neurons and also on iPSC neurons, and hopefully convince you that there's a value, a translational value in having these more physiological relevant assays. And this is something that we hope and envision in the future will then be used in disease modeling and also in drug discovery. This is just around the corner, I think, whereas later down the line, maybe precision medicine, so personalized medicine could also be something this would be applied on. This is what I brought for you today, so I want to say thank you, and I would like to take any questions.
Thank you for a really nice talk. I'm just wondering, with the initial disease that you talked about, the was it the SMA? Yeah. Is it more prevalent in certain sexes? Is it like more prevalent, for example, in males? And if so, do you consider looking at neurons which are derived from the different sexes and to see the activity of the channel you were targeting?
That is, we haven't done it, but I think it will be super relevant. So I believe it's this is from males, so both the control and the disease is from male. But I think you're right. We're often forgetting the women. That should also be included in the study. Yeah, thank you.
Thank you.
Hi there. There are a lot of different over this side. There are a lot of different TMEM channels, obviously, and they're quite a diverse group overall.
Yeah.
Can you tell us a little bit more about the TMEM one you just talked about?
There's very little known about it, and I'm afraid I also need to refer to Tabea if you want to know more about it. It's never been shown to be interacting with sodium channels before. So it is the first time we've seen this, at least. Yeah.
Okay, time to go to the poster. Thank you.
Yeah.
Thank you. I just have a question regarding the muscular dystrophy.
Yeah.
You didn't show the effect of the drug on the control.
No, we
neurons.
didn't.
And
There wasn't any effect.
that have any
Yeah,
effect?
there wasn't
so
any effect, yeah.
what is the reason of this upregulated sodium channel current?
I can turn that around and say we don't know why it's upregulated, but with the disease model here, then we can at least show that the phenotype of the neurons,
Okay.
that you have this excitability and increased ion channel function. I mean, there's no locally there's no known connection to the ion channels yet, at least.
Okay.
Yeah.
Okay. Thank you.
This was actually my question. Another question came to me. If you compare different iPS cells from different controls or from different patients, how variable is the current density of the sodium current?
Ooh, that's a really good question. Because I think we're lucky in this case because often we see that there's a larger person-to-person variation than that within the disease and disease corrected cell type. So therefore, you don't see it's really difficult to find somebody you can compare to. Then it should be a twin or a family member, but still, the difference between the people are often larger than between the diseased person and then the treated cells. So it's getting more used to you need to have the cells for the mutant, and then you have to CRISPR correct this mutant, then you have the healthy control, and then you CRISPR change this healthy control having the mutation, and then you can start to compare. Here we're really lucky that it just showed what you want to see, but I think this is the correct way of doing it if you want to go down that line.
Thank you very much. So, I think in the first topic, so you measure the action potential in the dissociated round neuron, but I think the subcellular localization of ion channels are strictly regulated in neurons. So, what do you think, or do you have any idea to extend the APC method technique to measure neurons with processes? What do you think about the meaning of evaluating the neurons without processes?
Yeah, you're right. You lose some information by losing the processes, and I think that's a cost going into high throughput. Then you will lose this. It would be super interesting to culture the cells into the chip. We have never done that. So, but if you're interested, we can try. It's so you can look into what changes that will do to the system. Thank you.
Hi. My question is about hippocampal cultures.
Yeah.
How do you deal with the fact that there are so many different neurons in hippocampus, pyramidal cells, interneurons, all kind of types? Does it influence your results?
Of course it does, and we don't separate them, so it will just be the entire pool of neurons, and it is enriched, but it's maybe not pure, so there might also be some astrocytes there. So this is also why we have tried to do magnetic labeling, and we also have a cell sorter, but then you need some sort of tag to sort the cells. But then you go specifically for one cell type, if that's what you want.